Hi, this is Antonio Sparra from the University of Bologna, and I'd like to introduce you to some experiments that we performed to evaluate the efficacy of two Selleckchem inhibitors directed to different types of cell cycle kinases, aurora kinases and polo-like kinases.
What do we work on? We use cell lines as a model of T-cell acute lymphoblastic leukemia, which is in many cases a deadly pathology for both children and adults. In this study, we used several cell lines with different features to evaluate the in vitro activity of the polo-like kinase 1 inhibitor ON01910 and the pan-aurora inhibitor CCT137690, either as single drug treatment or in combination.
And why targeting protein kinases for cancer therapy?
Actually, many mechanisms that support tumor cell hallmarks, such as invasion, limitless replication, growth, angiogenesis, death signal evasion, and others, are mediated directly or indirectly by a myriad of protein kinases. Nonetheless, the novel combinatorial chemistry techniques are able to generate a multitude of compounds able to interact with almost any protein within a cell. Therefore, the mission of biological laboratories is to find reliable and high-throughput techniques to quickly identify the compounds that best interfere with the desired target. Polo-like kinase 1 and aurora kinases are our targets, which are inhibited by two different chemicals provided by Selleck. In order to pharmacologically characterize our test compounds, we first assess an IC50 by MTT assays, and then we try to confirm the specificity of their action in cell cycle analysis by flow cytometry and by evaluating the phosphorylation status of some of polo-like kinase 1 and aurora kinases targets. An important aspect that we wanted to discern was whether these two drugs could have a synergistic effect on leukemia cell proliferation. In fact, due to the variety of factors that support cancer cell survival, the use of multiple drugs directed at different targets at the same time might prove beneficial. We must clearly bear in mind, though, that there might be an unpredictable interaction effect that should be profoundly examined before advancing with clinical trials. The clear advantage of drug combination is the possibility to lower the dose of the single drug, which results in lower toxicity and a better ability to withstand relapse and drug resistance, because it is more difficult for a cancer cell to develop simultaneous resistance toward two different mechanisms. Modern pharmacology provides several softwares by which it is possible to assess a combination index of a co-treatment. A more classical approach is based on the so-called shift to the left of the sigmoidal curve to a lower IC50 when the drugs are administered together. But let's move to the bench. In our assays, ON01910 demonstrated a good capability to inhibit cell growth in a concentration-dependent manner, and its calculated IC50 mean was around 516 nanomolar. On the contrary, CCT137690 was less able to do so, and the measured IC50 mean was around 8.3 micromolar. In our experiments, vincristine was used as a standard. Then we tried to combine the two drugs in a constant ratio, and in this case, CCT137690 could somehow redeem itself, showing an excellent ability to synergize with ON01910, as shown in the combination indexes table on the right. Also in the graph, there is a visible shift of the inhibition curve toward the left, and the measured IC50 of the combination was significantly lower than the one obtained by single drug treatment. In order to obtain a higher content analysis, we then tested the effect of the drugs on the cell cycle. Actually, while cyclin-dependent kinases are the driving force for cell cycle progression, polo-like kinases and aurora kinases exert a fine spatiotemporal control of the cell cycle events, with their mission being to maintain the fidelity of cell division. A malfunction of these proteins normally results in a mitotic block, for the cell is not able to overcome the cell cycle checkpoint. And this is what we expected to see by treating leukemia cells for 48 hours with both of the inhibitors. And as you can see, our results clearly show the ability of both of the drugs to induce a very significant G2M block, with the appearance of a sub-G0 peak indicative of the presence of apoptotic cells. Finally, we wanted to be sure that the drugs could be able to specifically block polo-like and aurora kinases, and to do so, we assessed the phosphorylation status of some of their targets. This proof is very important, because protein kinases, which play an important role in the cellular interwoven signaling network, phosphorylate some specific targets, which can then be exploited as pharmacodynamic markers. In our case, we chose two known polo-like and aurora kinases substrates, such as histone H3 and transformic acidic coil containing protein 3. We tested as well whether the treatment could influence the phosphorylation status of the two proteins. It is apparent that we didn't see any significant variations in polo-like kinase phosphorylation status before and after treatment, though it looks like there is an accumulation of the phosphorylated form of aurora kinase B, which might be a consequence of the mitotic block as assessed by flow cytometry. Pharmacodynamic markers show a clearer view. Both drugs demonstrated to have a specific action, which resulted in a reduced phosphorylation of TEC3 and HH3, which are reported substrate of both aurora and polo-like kinases. In conclusion, we found that the parallel inhibition of aurora and polo-like kinases has a synergistic effect in blocking leukemia cells proliferation. Both of the drugs tested, ON01910 and CCT137690, demonstrated to be selective toward their targets and able to cause a G2M block in our in vitro experiments, with a reduction in cell proliferation, at least to some extent. Finally, I would like to mention and thank Selleckchem again, which provided the drugs for our tests, which proved to be very reliable due to their excellent purity and quality of synthesis. In summary, we tested ON01910 and CCT137690. For all information about them, I will suggest you to visit the internet address www.selleckchem.com.
Thank you for your attention.
For any further question, please feel free to contact me. Bye.